Hi everyone, this is the Scrutinizer aka Dr. Noll. COVID-19 vaccines, that's what I want to discuss. Since their release, there has been arguments all over the place. On both the sides, one screams the vaccines are 100% safe and effective. That is the COVID-19 vaccine specifically. And the other screams we are becoming human lab rats. The question is, who is right? Louis Pasteur had said, science knows no country because knowledge belongs to the humanity and is the torch which illuminates the world. Therefore, do not let yourself be tainted with a barren skepticism. So let's look at the data and find the truth for ourselves. I've spoken actively to the anti-vaxxers in the past regarding the classic vaccines that have eradicated horrible diseases in all parts of the world, providing proof to show them how important vaccination is in keeping the eradicated diseases away. So now we have for the first time genetic vaccines that were approved without full and thorough research due to the pandemic. Just because the new COVID vaccines are labeled with the name vaccine and have been used by the masses, it does not mean that they don't have adverse reactions or are fully studied or can be deemed 100% safe and effective. That's the reason the whole system of clinical trials is in place to ensure that we have thoroughly checked the drug or vaccine for efficacy and the side effects. So throughout this week, I'm going to go in depth about the scientific concerns about the COVID-19 DNA and mRNA vaccines. These are not opinions, not controversies, and definitely not conspiracy theories. These are real and valid scientific concerns. To date, the companies, Moderna, Pfizer, J&J, they all have been trying to get mRNA and DNA vaccines approved for various diseases like SARS, MERS, HIV, Ebola, long before COVID-19 came into the picture. Yet now, within 3 months, they have managed to produce a vaccine that is 100% safe and effective. So here lies the problem. Just because we don't know the negative effects of a vaccine doesn't mean they are not there. We can't have just wishful thinking and save lives. What do I mean by this? Today, I'm specifically discussing the mechanism of the DNA vaccines that explain the possibility of reported adverse events. The DNA vaccines we have are from Johnson&Johnson and AstraZeneca, that is the ChAdOx1 vaccine. If you want to get the background information on what these vaccines are, please check out my previous video where I have explained all the vaccines in depth and compared them as well. And there is a link in the description box. So I am taking the example of replication deficient chimpanzee adenovirus vectored vaccine. That's the ChAdOx1. And it encodes the full-length wild-type SARS-CoV-2 spike protein. It also includes the transmembrane domains so that it is trafficked to the cell surface with a tissue plasminogen activator signal sequence. Understanding the molecular features of the expressed viral antigen is important for the interpretation of the immune response to this vaccine. We are told that DNA will go in the cell, make a little spike protein, and then immune system will get activated and destroy the protein, and we are protected. Is it really happening like that? I did not come across any references where someone has studied the phenomena and confirmed it. Rather, I have come across important details regarding the mechanism of DNA vaccines that I'm presenting here in the hope that there could be an input from scientists and non-scientists alike, and we could have a better understanding of this vaccine to benefit us all. So like I always say, please listen to my facts with open mind and then check and make a conclusion with your own critical thinking. I'm not asking you to trust me or believe. So the questions we need to ask are, when the authorities like CDC, FDA, etc., they say that a little spike protein will be formed in our body with the COVID-19 vaccine, what does that mean? Little could be 1 mole of the protein, 1 picomole of the protein. It's all relative, but the point is our body is not used to making or having a functional spike protein. Secondly, this protein is a structural protein, which means when it's formed, it needs to attach somewhere. When it attaches to any cells, what are the consequences? It has been shown that ChAd-OX1 gives a strong immune response. So here the question is, how much of that immune response is from the vector alone, or the antigen alone, which is the spike protein, or is that the immune response, what they're saying, is a combination of both? However, we need to know how much of that is a specific immune response to our spike protein. So here's the moment of the truth. The reality is that with DNA and mRNA vaccines, we are in a completely new and unknown territory. Why am I saying that? Of course, as a geneticist, it is a really exciting area, and to see that in fact we have figured out a way to synthesize the required proteins in the body using mRNA or DNA technology. In case of COVID-19, we are making the spike protein, which is a foreign protein for us humans, thus being used as an antigen to elicit a targeted immune response. Theoretically, the idea is great that our immune system will get activated and destroy the protein. Especially, it will generate the required immune cells with memory so that we are protected against the virus. Is this the way the endogenous system is really working? So we know it takes about 2 weeks at least for our immune system to be activated against the foreign antigen, in this case the spike protein. In the meantime, we have produced the spike protein, as I mentioned before, is a structural protein, which means the S protein is a trimeric class 1 fusion protein. It is composed of 2 subunits and they are functional. So S1 subunit, which is responsible for receptor binding, and S2, which is responsible for the membrane fusion. So the surface of the envelope spike is dominated by host-derived glycans, with each trimer displaying 66 N-linked glycosylation sites. This also means it's just not floating freely in the circulation, but attaches to the first cells it sees, and those cells would get glycosylated or glycolated. However, in this case, it's both glycosylated and glycolated. So first of all, because it's not part of the human system, does not have a specific function, does really not know where to go. So it does what it knows from being part of the COVID virus. It attaches to any cells that comes in contact with it. So since our vaccine ChAd-OX1 is given as an intramuscular injection, the spike protein will attach to the muscle cells like fibroblasts and also immune cell types or other cells that it might go to. But the peculiar difference is that these cells, the muscles, the fibroblasts, the immune cell types, they are not rich in ACE2 receptor. Spike protein, as we all know, has been shown to attach to only to the cells that express high levels of ACE2. For example, the nasal mucosa, GI tracts, lungs, heart, etc. This has been shown recently in a peer-reviewed publication that has done a thorough research study taking into consideration all the right controls and asking the important questions. Like Einstein said, a scientist is not one who has all the right answers. However, it's a person who asks the right questions. So the study that I'm mentioning, it showed that ChAd-OX1 attached to the cells it came in contact. So this group, they use both in vitro and in vivo mouse model. The sera of the vaccinated mice showed about 60 to 70% of the cells expressing S protein in the mice, and the presence of the S glycoprotein was also detected at the cell surface of the infected HeLa cells, and these are the lung cells, so in vitro, in a petri plate. Similar to the native viral infection, the cells infected with ChAd-Ox also shed their S1 subunit. Now, whether this is a beneficial or detrimental feature with respect to elicitation of immune responses during vaccination, that is unknown. Meaning, it was important to have this answer before we started mass vaccination. Shedding of S1 subunits from viruses, they occur during native infection, and ChAd-Ox1 derived S protein mimics this native feature of the viral spike protein. Secondly, spike protein is a heavily glycosylated protein, meaning when it attaches to any cells, they become heavily glycosylated or glycolated. This has been confirmed by tomograms. Again, this data is presented in the research paper I just mentioned, and I will leave the reference in the description box also. So the confirmation came by tomograms, which showed that the surface of the cells is densely covered with protruding densities consistent with the size and shape of the pre-fusion conformation of SARS-CoV-2 S protein. These densities were absent in the control uninfected cells. So, meaning that they were specific. So, furthermore, high levels of glycan occupancy were also observed across the entire S protein. And this is especially important since glycans are known to shield more immunogenic protein epitopes. And these host-derived glycans mask immunogenic protein epitopes from humoral immune system. It's a common strategy that is utilized by the viruses. So the point I'm trying to make is that our muscle cells are not used to being heavily glycolated, let alone the spikes coming out of them. And this apparently has to have consequences. For example, fibroblasts maintain a structural framework for many tissues, and overglycosylation could have detrimental effect on our muscle cells or cause aging, etc. The pathological role of excessive glycation is evident in various types of disorders and is published, such as diabetic vascular complications, neurodegenerative diseases, melanoma, growth, metastasis, and so forth. Then the biological effects of excessive glycosylation include inactivation of enzymes, cross-linking of glycosylated proteins, trapping of the soluble proteins, abnormalities of nucleic acid function, altered macromolecular recognition and endocytosis, and increased immunogenicity, to name a few. So the analysis from the research paper also revealed a presence of abundant pre-fusion spikes on the cell surface, while little post-fusion spikes were detected. This is of interest and in contrast with the mRNA vaccine. So I'll explain this phenomena in the next video where I cover the mRNA vaccines. So these details could explain the reported adverse events after the ChAd-Ox1 vaccine, like the thrombotic thrombocytopenia, where the blood clots form in small blood vessels throughout the body. The neurological symptoms such as paralysis, paresthesia, or allergy-like reactions. Some of these reactions, they were taken as allergy and treated as such. However, they were not the real allergic symptoms such as angioedema and foreign body sensation in the throat, which were unsolicited adverse reactions in the previous clinical trials. So in addition, beginning 5 to 16 days after vaccination, there is a report of patients, they presented with one or more thrombotic events, like cerebral venous thrombosis, pulmonary embolism, and other thromboses. Of these patients, 6 had died and 5 patients had disseminated intravascular coagulation. And 1 patient presented with fatal intracranial hemorrhage. And the median age of all these patients was about 36 years. So usually during a viral infection, the virus-specific antigens are present on the cell surface, and the cells release infectious virions by budding for a short period before the cell death. Thus, sometimes they are disseminated by the continuous spread from cell to cell without exposure to extracellular antibody. So, cell-mediated immune responses are believed to be important in controlling the local spread of this type of infection. The question here is that with the DNA vaccines, we have almost filled ourselves with the spike protein. How does that protect us from the real virus? Also, an immune response that's elicited does not mean we have generated memory cells, which is what we need from a good vaccine, so that when the real infection or the virus comes, our memory cells remember the antigen and are ready to fight off the specific infection. This immune response that we are seeing with the ChAd-Ox vaccine, as I suggest earlier, could solely be because of the adenoviral vector or combination of both the vector and the spike protein. So therefore, to make a vaccine without thinking of all the consequences is highly irresponsible, especially during a pandemic when people are scared and will believe anything that the authorities tell them. How many times CDC has changed mandates? In addition, when any adverse effects are presented, to say that they are not related to the vaccine or that the number of people is so small that it's all right to have those adverse reactions or even death, it is simply callous attitude. If we look at our classic vaccines, example against MMR, they did not kill any of the people with adverse events. Usually, a vaccine does not produce adverse events that could induce death in the people or any of the long-term chronic illness. So the other thing I need to mention is that everyone is still going on the hypothesis that COVID-19 is a respiratory disease. The symptomology is all over the place, from respiratory system to neurological and to GI system. And then we have asymptomatic people as well. This tells us it is an endothelial or a vascular disease. Lastly, a very recent publication has shown and confirmed that the spike protein itself, it contributes to damaging the vascular endothelial cells. That is, the spike protein alone is enough to cause inflammation in endothelial cell lining the pulmonary artery walls, thus damaging the lungs and arteries. This is, I have to admit, a very, very scary discovery and a game changer. And I will cover it at another time so I can explain this whole article or the mechanism. So now, all the more reason to have done a thorough clinical trial and looked at every reported adverse event. In addition, the vaccine manufacturers should have looked at their own previous failed vaccines. That is it possible that spike protein is not the right target for making this vaccine? I'm saying this because COVID-19 spike protein is even more complicated than SARS. Once again, I'll have to explain the structure of this spike protein or compare it with SARS and MERS at another time. My whole discussion is not about blaming someone or something. Rather, as I always say, the good science means when we find a shortcoming in our data or the released products, we have to go back and revisit our mechanism or the experiments and so forth. And I'm not seeing that happening. In conclusion, it seems most of the people and the authorities are defending the vaccine's adverse effects rather than regrouping and saying, what can we do differently? So we have an effective vaccine. Side effects are okay, but they are the least side effects. For example, if we compare with our classic vaccines. So like Einstein said, doing the same thing over and over again and expecting a different result is plain stupidity. So what is the take-home message from this? Keeping in mind the new research data I presented and the valid concerns regarding the DNA vaccines for COVID-19, these vaccines should not be mandatory for the entire population, especially children, teens, college kids. Similarly, there should be no COVID-19 passport because we will find out more adverse events based on the data I presented. Why should entire population be vaccinated blindly and then cause chronic health problems in the population? Third, herd immunity is not happening or can't happen because these vaccines right now are not capable of giving the immunity that we need to create the herd immunity. Once again, I have explained it in a future video. Therefore, we need to be very careful in implementing these strict rules regarding COVID-19 vaccination. We all know there is no returning to the past. So I will place the list of videos that I'll release soon in the comment box along with their dates, so that if you're interested, you could check back for further details. Or if you like this video or you're interested in knowing more, you could also subscribe this channel to be notified of the new videos when they are posted. And as I always say, think critically, stay healthy. The Scrutinizer out.